Hello friends, Ace Botany next episode. In this episode, we'll be discussing Cell, the Unit of Life, chapter number 8, episode 6. So what all things we'll be discussing in this episode? For that, I will share the screen so it will be convenient for you. I think screen came, so we can proceed. Chapter 8, Cell, the Unit of Life, episode number 6. Theoretical chapter, important for theory exam as well as NEET exam. So topics, as usual, there will be an introduction. Then eukaryotic structures, cilia and flagella, centrosome and centriole, microbodies, cytoskeletal structures, and after that, 5 theory, 5 NEET model question with the key. And remember, in shorts, I am daily putting 3 NEET model questions. So to get all this, subscribe Ace Botany. Okay, we can proceed. Introduction. So basically, we are continuing with the eukaryotic cell structures. And in this episode, cilia, flagella, centrosome, centriole, cytoskeleton, microbodies, then questions. Now we can proceed to cilia and flagella. So NCERT diagram, cilia, flagella. It is a labeling question. So we can proceed with it. Cilia, I think first page is clear. We can proceed to second page. Cilium, that is originally singular. Flagellum, also singular. And they are normally fine hair-like outgrowth from cell membrane or connected with the cell membrane. And cilia is small and many in number. Work like oars. Spelling is O-A-R-S, oars. Flagella longer and limited in number. Movement of cilia can cause movement of either cell or surrounding fluid because many cilia will be working. Flagellar movement can cause cell movement. And remember, bacteria also have flagella that we studied in prokaryote, but different from eukaryotic flagella, including structural aspect. And these two diagrams, again, old NCERT showing a power stroke and recovery stroke in connection with the movement. How the structures works. I think second page is clear. Now we can proceed to third page, where we'll be discussing differences between prokaryotic flagella and eukaryotic flagella. Many are there. I just represented 4 or 5. In prokaryote, protein is flagellin, while in eukaryotic flagella it is tubulin or dynein. Prokaryotic flagella 9+2 arrangement is absent, while other case 9+2 organization. Now prokaryotic flagella not covered by any sheath, while in eukaryotic plasma membrane derived sheath is there. Then prokaryotic flagella it is single-stranded, other side it is 11-stranded. Prokaryotic it is single, eukaryotic it is 11-stranded, 9+2. Now flagella rootlets are absent, while other case that is eukaryote present with the supporting function. So some basic differences, few, 8 or 10 are available. Few I just represented. Now comes structure. See, both the cilia and flagella are, indirectly we can say, extensions of cell membrane or connected with the cell membrane. Membrane-bound structures. Normally, they arise from a structure called basal body, and the arrangement is 9+0, like centriole. So 9+0 arrangement. Core of cilia or flagella is called axoneme. So core of cilia or flagella is called axoneme. Up to that, I think it is clear. This is a diagram, labeling question. Now, axoneme. We can proceed to next page. Few more diagrams are represented. Here you can find. Rootlets, etc. we can find. As old NCERT diagram, present NCERT, such diagrams not there. So we can continue. So axoneme, which has got number of microtubules running parallel to long axis, Axoneme is the core of cilia and flagella. Axoneme is made up of 9 microtubule doublets. Radially arranged along the periphery diagram we can find. Along the periphery, there is a pair of microtubule at the center. It is present singly here. A pair is there but existing singly. So total 9 doublets plus 2, that way. I think idea clear, diagram clear. Now one more set. This is old NCERT diagram. This is new NCERT diagram. Just a comparison. Here more labeling is there. Here present NCERT. Better What is the new edition NCERT? Just of this I just given for a comparison or reference only. This is our latest diagram. Latest diagram NCERT, better pass through. It's only for comparison. Now, tubules at the center is connected. Here you can find, say, tubules at the center is connected to each other by a bridge and enclosed by a central sheath. Here you can find central sheath. Central sheath also connected to peripheral doublets or doublets by radial spoke. These are the radial spokes. It's very easy. Diagram is there, very easy for you. So central sheath also connected to peripheral doublet by radial spokes. That all are microtubules. 9 radial doublets, 9 radial, this way 9. So 9 radial doublets, thus 9 radial spokes. See each doublet, radial spokes. The outer doublets also connected to each other by a protein called nexin. See this way. There is a connection. Each other by protein called nexin. And there is a plasma membrane at outer. Here you can find. I think idea clear. Doublets that is named as A and B. That also will be connected. That's regarding structure. Now we can proceed to next page. Some differences between cilia and flagella. Theory question. Cilia, their number per cell is more. Flagella limited. Cilia smaller, flagella longer. Cilia seen throughout, flagella normally at one end. Cilia move in a coordinated manner, connected with the locomotion, feeding, aeration, circulation. While flagella normally independent. Limited number will be the independent and helps in locomotion. That is regarding cilia flagella. Now comes the next structure, centrosome and centriole. All these old NCERT. Just shown it, that's all. Centriole. The present NCERT, I think no diagram in NCERT. Just to show old NCERT diagrams. Now we can proceed to next one. So when we come to next one in the sense, next page, centrosome and centriole. So what are centrosomes? Centrosome is an organelle usually containing 2 cylindrical structures, centrioles, which are surrounded by an amorphous pericentriolar material called centrosphere or kinoplasm. Diagram will be coming, so it will be clear for you. Kinoplasm. I think up to that it is clear. Now diplosome. Theory and question. Diplosome means diplo 2, so body. So 2 centrioles are called diplosomes seen in centrosome, arranged in right angles to each other. Now comes organization of centriole. Show the diagram. This is not in NCERT, just drawn it for your reference. Organization of centriole, which is otherwise called cartwheel-like arrangement. It is basically 9 plus 0 like basal body. Centriole also we can find 9 peripheral fibrils of tubulin, evenly placed, but at the center it is absent. Center it is absent, that is why it is 9 plus 0. Cilia flagella 9 plus 2, here it is 0. Fibrils are made up of 3 subfibers. Here you can find 3 subfibers. Term is triplet fibrils. Having subfibers, it is labeled there A, B, C. And here you can find C and A will be connected here. CA linkers, CA connection. So there is CA proteinaceous linkers connecting the adjacent triplet fibrils. There is a central hub region. This is the famous hub region. Famous hub region. It is having rod shape and chemically proteinaceous in nature. From the central hub region, 9 radial spokes here. 1, 2, 3, 4, 5, that way. 9 radial spokes, proteinaceous, are developed towards the periphery to triplet fibrils. See this way. This way. And that gives a cartwheel-like arrangement. Centriole is a membranous structure, no membrane. It have 9 peripheral massules. These are massules. 9 will be the massules or pericentriolar satellite. Short form it is called MTG or microtubular generator. Centriole is absent in higher plants. Higher plants it is absent. Seen in animal cells, algae, fungi. And cell cycle we studied, centriole duplicates in S shape of cell cycle in animal cell. Centriole. Why animal cell? Centriole is predominantly seen in animal cell. Now, what are functions of centrioles? When we come to centriole, linked with the basal body transformation, a link is there between basal body and centriole, which in their turn gives rise to flagella and cilia. Connected with the cell division by spindle formation, spindle apparatus, cell cycle we will be studying again, already studied. Now comes microbodies. NCERT given a definition for microbodies, many membrane-bound minute vesicles called microbodies. They contain various enzymes and are present in both the plants and animals. Now we are discussing eukaryotes only. NCERT page number 102. Under this microbodies, three structures are referred in NCERT. One is peroxisome, other is spirosome, third is glyoxysome. So we'd expand it, peroxisome. Name is due to presence of enzymes for peroxide biosynthesis. They are present in plants and animal cell. In plants, photorespiration, you'll be studying in photosynthesis, photorespiration or cycle. Photorespiration is connected with the peroxisome. And two famous enzymes. One is glycolate oxidase. It is concerned with the production of peroxide. Other is enzyme catalase, destroying peroxide. Now, second microbody is spirosome, connected with the storage and synthesis of fats, commonly seen in oil seeds, endosperm cells, and they have hydrolytic enzymes, which is otherwise called even spirosome, sometimes refers to a plant lysosome. And scientists believe that is SER produces or connected with the spirosome production. Now comes glyoxysome, third microbody from endoplasmic reticulum vesicles. That is what scientists suggest. Glyoxysomes will be formed. Endoplasmic reticulum vesicles. Indirectly it is formed. Glyoxysome arises. And they have enzymes for glyoxylate cycle. What is meant by that? You may study in zoology still, fat to carbohydrates, as in germinating fatty seeds. And in the case of animals, this conversion is not possible because glyoxylate enzyme is absent in the previous NEET exam question. So animal side, this conversion, fat carbohydrate conversion is not possible because glyoxylate enzyme is absent in them. Now comes last structure, cytoskeletal structures. So what is cytoskeletal structures? It is an elaborate network of very minute proteinaceous in the sense fibrous, filamentous, and tubular structure. It forms a structural framework inside the cell and present only in eukaryotic cell. They are involved in many functions like mechanical support, motility, maintenance of the shape of the cell division, etc. Here also three structures will be there. Microtubules, microfilaments, intermediate filaments. Now comes microtubules. They are unbranched and hollow, made with the protein tubules, tubulin, microtubule, that we can remember. Calmodulin, a calcium binding protein, is important for assembly of microtubules, and they have indefinite growth. The wall is formed of 13 protofilaments, 13 laterally associated and helically arranged longitudinal strands. And they are having alternate spirals with the alpha tubulin and beta tubulin. Assembly and disassembly of microtubules requires calcium Ca2 plus and GTP. And cell cycle, you may study a mitotic poison, colchicine. Colchicine can prevent microtubule spindle formation. Now, next cytoskeletal structure. Now, functions we have to discuss. Functions for microtubules. Some important functions include connected with this. Many functions are there for each cell organ. Mainly, what connected with the NCERT, we are just proceeding. Connected with the cell division, spindles and astral rays. Connected with the cilia flagella, cell motility shape, etc. Then movement of chromosome in anaphase, connected with the cell plate. Now comes next cytoskeletal structure, that is microfilament. Again, long and narrow, cylindrical in shape, made with the protein actin. Solid. Actin is a globular protein, have myosin filament proteins also. So both are involved. Okay, so made with the protein actin. Microfilament normally is solid. Actin is a globular protein and myosin filamentous protein is also connected. Now, functions of microfilament connected with the cytoplasmic stemming or cyclosis, helping plasma membrane in various manner. Pseudopodia formation associated with the cleavage furrow. Microtubule is cell plate. Microfilament is cleavage furrow. Cell division will be studied. Now, intermediate filament. They are frequently formed a network, non-contractile, chemically acidic, some functions only because many will be there or many are there. Doing mechanical functions, In most of the animal cells, they are involved in formation of, next is important, scaffolds for chromatin and forms a basket around the nucleus. So pure theoretical aspect, but frequently comes for theory and entrance. Now after that, 5 theory question, 5 NEET model question with the key. So what all things we studied in this episode? We can just rewind it. So you'll be getting a continuation what all things we learned in this episode. So cell, the unit of life, chapter number 8, 6, introduction, what all things we'll be learning. Then cilia flagella, cilium flagellum. Then from where it grows, connected with the cell membrane, difference between cilia and flagella, and cilia moves like an O, O-A-R-S-O. Flagella, I told you, and flagella longer and limited in number. Movement of cilia can cause movement of either cell or surrounding fluid. Flagella movement can cause cell movement. Then bacterial flagella and eukaryotic flagella, different in many aspects. This too shows that power stroke and recovery stroke. Then differences between bacterial flagella and prokaryotic, eukaryotic flagella. Then structure, that is basal body 9+0. Then core is axonym, which has got, so 2 sets of diagram. One is here, previous page also. Number of microtubules running parallel to long axis. Then periphery 9 microtubular doublets, readily arranged. Then there is a pair of microtubule at the center, present singly, so totally 9+2 arrangement. Basal body 9+0, centriole 9+0. Now again, 2 diagrams, old and new NCERT comparison. Then structure explain, 9 radial spokes, 9 radial doublets, so totally 9 radial spokes. Then doublets are connected to each other, like AB, you can find in the diagram. A and B of A of one will be connected with the B of the other. That way it is linked. Then next in protein is the plasma membrane outer side. Then differences between bacterial flagella, difference between cilia and flagella, difference length, etc. Then next is centrosome or centriole old NCERT diagram. Then usually containing centrosome. What is centrosome? It differs from centriole. That is centrosphere or kinoplasm. With the centriole, centrosome. Then diplosome to centriole. Then organization of centriole 9+0, which is otherwise called cartwheel arrangement, like basal body. Then 9 peripheral fibrils of tubulin evenly placed, but at the center it is absent. Cilia flagella, center 2 are there. Fibrils made up of 3 subfibers, A, B, C. CA linkage will be there. And there is a central hub region also we can find. It is having rod shape and chemically proteinaceous. Then 9 radial spokes. Then the proteinaceous are developed towards the periphery to triplet fibrils, cartwheel-like arrangement. Centriole is membranous. Then it has got 9 peripheral massules, pericentriole or satellite or MTG. Centriole absent in higher plants, seen in animal cell, algae, fungi. Centriole duplicate in S-phase of cell cycle in animal cell. And functions of centriole, that is basal, linked with the basal body formation, which in their turn gives rise to flagella, cilia, connected with the cell division, spindle formation, spindle apparatus, etc. Then microbodies. So when we come to microbody, many membrane-bound minute vesicle called microbody, present in both plants and animals, NCERT page number 1 or 2. Then peroxisome, spirosome, glyoxysome. Then why the name peroxisome and what is their function? How they involved in photorespiration? Enzyme, 2 famous enzyme, glycolate oxidase, catalase, what is their function? Then spirosome, connected with the storage and synthesis of fat, where we can find oil seed endosperm cell, pyrolytic enzymes, often called plant lysosome. Scientists believe SCR is producing spirosome, then glyoxysome, connected with the endoplasmic reticulum vesicles. Then enzymes for glyoxylate cycle, fat to carbohydrate, as in germinating fatty acids. Animal cell conversion is not possible because glyoxylate enzyme is absent in them. Then comes last part, cytoskeletal structures. What is meant by cytoskeletal structure? What is their function? And 3 famous cytoskeletal structures: microtubules, microfilaments, intermediate filament. And what is the structure of microtubules? That is tubulin, 13 protofilaments, alpha tubulin, beta tubulin, then calcium and GTP. Colchicine can prevent microtubule spindle formation, mitotic poison. They act on microtubule. Now, functions of microtubule, cell division, then cilia flagella, cell motility, shape, then movement of chromosomal anaphase connected with cell plate. Then next is microfilament. When we come to microfilament, long and narrow, cylindrical, made with actin, solid. Actin is globular protein, myosin is filamentous protein. Function, cytoplasmic stemming cyclosis, helping in plasma membrane in various manners, pseudopodia formation associated with the cleavage furrow in animal cell, cell plate in plant cell. Intermediate filament, frequently formed network, non-contractile, chemically acidic, some functions, mechanical functions. Most of the animal cells, they are involved in formation of scaffolds for chromatin and forms a basket around nucleus. That is important. Then after that, 5 theory question, 5 NEET model question with the key. Remember, daily I am putting 3 shots. One will be around afternoon time, 12 to 1:30, etc. Then after that, 2 more NEET type question, that is around 8:30 to 9:30 time. So we can meet later. So if you like this episode, subscribe or like it, share it, comment on it. Take care. Bye. Thank you.